Hello children, I hope you all are fine. In this video, we are going to discuss about antiviral drugs. A virus is a microscopic organism which can only be observed with the help of an electron microscope. A virus is composed of a genome, DNA or RNA, which is wrapped inside a protective protein coat which is called as capsid. Children, most of the viruses have an extra envelope made of lipid with protein spikes that serve to attach to the host cell. Children, viruses are actually an intermediate form between the living and non-living. They are acellular and do not have a nucleus in them. Viruses can breed only within the cells of other organisms, that is the host cell, and they cause specific diseases in specific hosts. Virus enters inside the host cell after getting attached to the membrane of the host cell. Inside the host cell, they use host cell machinery to multiply themselves. As antibiotics can't harm the human cell, they are also ineffective against viruses because viruses affect the human cells by entering into them. Unnecessary intake of antibiotics during viral infection weakens the immunity system of our body because these antibiotics will then attack the natural flora of the body, and many good bacteria which are present in our body will be killed in this process. These bacteria are actually very helpful in maintaining the immunity system of our body. So, to fight against the viruses, there are special compounds called as antiviral drugs. These are specialized chemicals which, unlike antibiotics, do not inactivate or kill viruses, but they merely inhibit viral reproduction by interfering with a certain stage of the life cycle of virus. If we can inhibit any of this stage, we can stop virus from causing its infection. Antiviral drugs actually aim to block viral reproduction at any of these stages itself. Viral life cycle includes various stages. The first stage is the attachment of virus with a host cell membrane. Second stage is related with the entry of the virus inside the host cell. Third stage is all about the uncoating of the viral genome, that is release of viral genome. Fourth stage is related with the replication of the genome. Fifth stage is related with the protein synthesis in order to make protein envelope, that is capsid. Sixth stage is for assembling the viral protein, and the seventh stage is the release of viral particles. Different antiviral drugs function through different mechanisms. Some antiviral drugs binds with the spike proteins of the viruses, due to which viruses are not able to attach to the host cell membrane and thus not able to enter inside the host cell. Some antiviral drugs can attach to the genetic material of the viruses, DNA or RNA. This attachment makes virus to remain in the packed form and not release its genetic material inside the host cell, which makes it ineffective in causing diseases. Why is it difficult to make antiviral drugs? Let's see. It is difficult to make antiviral drugs because Viruses replicate entirely inside the host cell using the host cell's machinery, which makes it difficult to develop drugs that will affect viruses without harming the host cell.